So we're officially in exam 4 content. Wowza. So chapter 16, 17, 18 are our main focus right as of right now. So we'll start off with chapter 16, and then I'll briefly cover 17 and 18, just because the chapter 17 and 18 really just build off of chapter 16. So that would be the bulk of our focus is chapter 16. So the adaptive immune response, right? What makes it different than our innate, right? So our innate immune response is going to be very basic. It's going to be, this is foreign. That's what our body will tell us, is that this is foreign. We need to attack this, and we need to get over it. So that's going to be things like, your macrophages, right? They're just going to eat anything that's foreign. So that's going to be your innate, right? And then inflammation, fever. And like, don't even get me started on, your very, very first line, right? That's your intact skin, mucous membranes, and your normal flora. So with all of these things that are just kind of very general, at a certain point, if you have a pathogen that enters your system, it will get past those things. So once we break past the first line of defense, second line of defense, which are both non-specific and innate, we get to our third line of defense, which is adaptive. So the whole point of reaching this is that this is a big deal, and your body basically wants to learn what this is, so that if it ever comes across this again, you do not get nearly as sick, and it is trying to develop a memory. And if you did have this infection before, usually the memory cells will the cells will remember what this was and produce the correct antibodies. So the first stage of complement, first stage is complement. So to understand complement, for the sake of this class, you basically need to know the cascade. So we have our C1, which differentiates to C4 and C4A. And C4A just kind of goes out on its own. C4 then goes to C4B. C4B goes to C2 and C2B. C2 goes to C2A. C2A goes to C3. C3 goes to 3A, and that is what's responsible for inflammation, or that's part of what's responsible for inflammation. C3 goes to C3B. C3B goes to 3,5. 3,5 creates 3,5A, which is also part of inflammation. 3,5 goes to 3,5B. 3,5 goes to 3,6/7, and then that activates C5B and 6,7, and then we go to C8, and then at the very end we have C5B 6, 7, 8, 9, and that is our membrane attack complex. This is all very complicated, and I encourage you to go into the slides and review your complement system diagram, which basically just points all of this out. So all the complement proteins start with the letter C, as you know, and they are in the order that they were discovered, which means that they're not going to flow in order. But they usually circulate in an inactive form, and they are activated by splitting into smaller fragments, which is your C3A, C3B, and those are designated with that lowercase. And this is what we call a cascade, right? One is against the other, and which triggers the next one and another one. If you don't have the first one, you can't get to the next one. That is the goal of a cascade. And then there are 3 major outcomes of complement. But I'm going to take a quick break because somebody has arrived on Just kidding, they didn't. I guess they dipped. I don't know. But alas, there are three major outcomes of complement. It's opsonization, which is just enhancing phagocytosis. And this is specifically done by C3b. That's opsonization. Then our next outcome is inflammation. So when we have inflammation, it's increasing blood vessel permeability and chemotaxis. And it also attracts phagocytes. So we already made phagocytes more competent and more active, and now we're attracting them to the sites that we need them to be, and we're making the blood cells leaky enough to where they can go to the correct site. And then the last one is cytolysis. So this is where we burst a microbe, and when we burst this microbe, it's because we've basically attacked it and filled it with so much extracellular fluid that it just burst. And this attack complex, this is achieved by the attack complex, which is C5b, C6, C7, C8, and C9. That's the very end of it. That is our final blow to this bacteria. Understand that we first have C3, then we get C3a and C3b. Now C3b is going to continue to C3-5, but C3a goes off on its own to a mast cell, which releases histamine, which is usually responsible for inflammation. And so C3b now over here, it already cascaded down to C3-5. C3-5 has 3 feet. It has C5 has C5b and C5a. C5a does a similar thing that C3a did, which was, I don't know if I'm repeating these. So C5a does a similar thing as C3a, which is going to the mast cell, which releases histamine. But C5b, which is similar to C3b, goes down and activates C6. Now C6, we're starting that, the attack complex. So we have C5b, now we need 6, 7, 8, 9. That is usually the best way to remember it, is that 3 and 5 have these weird A's that go off to a mast cell, but everything else follows. And then our membrane attack complex is 6, 7, 8, 9, and then C5b is the weird one. And that's what our goal of cytolysis is. So C3b, right, is contributing to opsonization. And when C5a and C3a go off to that mast cell and release histamine, that's contributing to inflammation. And then C5b, 6, 7, 8, 9 are all responsible for cytolysis. So those are our 3 goals in action. So how do these parts actually contribute, and how do they reach their end goal, right? So some microbes can evade phagocytosis because they have a capsule. But the immune system can overcome this by coating a microbe with C3b, basically just giving it a handle, giving the immune system a handlebar. It gives the phagocyte something to grab onto, and it's not just like grasping at a very slick surface anymore. Now we have like an actual handlebar to have the phagocytes grab them, bring them in, and eat them. So that's how C3b, that's how it actually performs its job. Now, inflammation, it's pretty straightforward. C3a and C5a, basically what they do is they bind to the mast cell and cause the release of histamine. Histamine, as you know, increases blood vessel permeability, and it causes the inflammation to actually occur. So if you're thinking about an antihistamine, something like Benadryl, what do you take that for? You take that because you have allergies. So what happens when you have allergies, right? You're puffy, your throat might be scratchy and, swollen, but you are undergoing inflammation. And it's similar because when you actually have not just like allergies, but like an actual allergy to something like peanuts, then you will undergo basically extreme inflammation and anaphylaxis. So when you have inflammation of this degree, it becomes dangerous. But for the sake of complement, this is just enough to get the job done. We're not going overboard. And also remember that C5a attracts phagocytes. It's a chemoattractant. All right. And then cytolysis, we know that membrane attack complex, right? That's that's all you really need to know, is that it is going to rush all this extracellular fluid in and break it all open. So now we're going to move away from complement and towards a more adaptive immune system. So innate immune system, right? Let's compare and contrast again. It's present at birth, but it's not specific, and it's relatively quick. There's no memory, and those are things like your intact skin, phagocytes, NK cells, right? But if we look at our adaptive immune system, we usually get this over time, and it's through our exposure to certain diseases and bacteria, right? So it is not present at birth, right? We haven't really done anything. We haven't gotten an infection yet. We haven't been able to gain that memory. And it is specific. The response time is slow because of the process that a cell has to undergo in order for the adaptive immune system to be turned on. And we'll get into that a little bit later. But if you know what that is, good thinking. Just try to quiz yourself. So there is memory. That's kind of the whole point of the adaptive. And it's usually B and T cells and antibodies. Let's see. So there are 4 types of immunity, or acquired immunity. So we have 2 forks. So 2 and 2. There's the active. There's our active immunity, which is our bodies creating antibodies. And then we have our passive, which are usually just, somebody else had made them, and now they're from an outside source. So some of our active or our own antibodies could be something like, obviously the natural way, which is we get sick. We get sick with something, we get over it, and we develop the memory. Or we can get an artificial immunity. So artificially acquired active immunity could be something like an immunization. And I'm sure almost every one of us has had an immunization of some sort. So our naturally acquired active immunity is something like we get infected with something. Our artificially acquired active immunity would be something like an immunization. Now, our naturally acquired passive immunity, that would be something like when a mother breastfeeds, she can pass down her maternal antibodies to the child. Now, when we have passive, then that's a type of passive immunity. So an artificially acquired passive immunity would be something like antibodies from an outside source. So during this pandemic, we've seen a lot of plasma like transfusions and COVID antibody treatments, which work. So that would be something like that. Let's see. So we have 2 major components of the adaptive immune system. or adaptive immunity. We have the antigen and we have the antibodies. An antigen is from the pathogen, and it elicits the adaptive immune response. An antibody is the recognition and interaction with those antigens that eventually kill the pathogen. So antigens are mostly proteins or polysaccharides, but they recognize and react with antigenic determinants or epitopes. I don't want to say it wrong, but it's a weird word, so you just have to kind of memorize what it looks like. But they interact with this, and they are able to elicit that immune response. So usually the location of the antigens, remember antigens versus antibody, usually the location of the antigen is on the capsule, cell wall, flagella, fimbriae, or toxins of viral surface coats, and when antibodies come into contact with that, they usually bind. So yes. Another similar thing is haptens. They are molecules that are way too small to be antigens themselves, but they combine with carrier molecules. So they form this hapten carrier molecule complex, and it's now antigenic, and it stimulates an immune response. So let's say you have an allergen, right? An allergy to penicillin. So the penicillin would be an example of a hapten molecule. And a carrier molecule could be the serum and blood, and mix that together and you have an allergic reaction. Let's see. So to take a closer look at antibodies, they're globulin proteins. And that's why they're usually coined with the Ig, that's immunoglobulin. They're very soluble, they dissolve easily in the blood, that's why they can be carried so easily, and they are specific. They're specific to an antigen. And so this has at least 2 identical sites for binding antigens, known as antigen binding sites. It's pretty straightforward, but that's why they're usually depicted as a Y for the antibodies. You won't really need to know the antibody structure, but it is kind of interesting. You have your antigen binding sites, and the top is the heavy chain, the bottom is the light chain, and then you have your hinge region, and then your FC or stem region. And knowing the structure definitely lets you know how it bonds. But yes, let's see. So you have your 5 antibodies. You have your IgG, you have your IgA, you have your IgD, you have your IgE, and your IgM. Now, the main ways that we remember what these are is that, let's see. So IgG antibodies are a lot of the antibodies that circulate normally, and they are usually produced in large amounts. They are neutralizers. The IgA antibodies, they are usually by a dimer. They're a weird one. So the IgA antibodies, they're 2 antibodies, but they stick the bases of the Y's together. They're mostly in mucous membranes, not so much your blood. So they prevent attachment of pathogens to mucous membranes. That's why, we want our mucous membranes there in the first place, and that's why they're a part of the immune system. But yes, and then IgM, those are the mega ones. Those have 5 antibodies on a pentagon. But remember, they are monomer on B cells, and then they become a pentamer when secreted. So they have a non-secreted and a secreted version. but they can't move out of blood vessels very easily because of how big they are, and they're in fairly low percentage in the blood. And they appear first in response to a primary infection. But high levels indicate that there had been a recent infection. They respond to ABO blood group antigens. So if you were to be, transfused with the wrong blood type, this is what would come for your blood cells. And it enhances phagocytosis. It also promotes microbe aggregation and complement activation, which are both very good if you want to, get rid or clear any kind of infection. So if you want to know what microbe aggregation is, break it down. Microbe, right? We have a pathogen. Let's say it's just a bacteria. Aggregation. What's an aggregation? It's just a gathering. So if it promotes microbe aggregation, it promotes gathering all the bacteria into one place and then destroying them all in one fell swoop, right? It's much easier to hit one target rather than 30. So yes, and this is also the first antibody produced by a fetus. Okay, so IgE, we think allergy. This is literally, this is also a monomer similar to Igg, but they are in very, very low percent. It's mainly found in body fluid, skin, lymph nodes, mucosal tissues, not so much the blood. And it's, again, primarily the allergic reactions. And the FC regions bind tightly to mast cells and basophils. So the FC region isn't the antigen binding region. It's going to be that base. When it binds, it causes mast cells or basophils, whichever it's bound to release histamines and cause allergic reactions. This attracts complement, it attracts phagocytes, and it can be protective, but usually if it's over the top, that's when we get anaphylaxis. This can be in response to parasitic worms, and we can use this as a diagnostic of certain worms. Let's see, IgD. So IgD is another monomer, just like IgG and IgE, and it's also in very low percentage, and it's mostly found on the surface of B cells, but it's rarely secreted. So similar to IgM, they're on the surface of B cells, but they're rarely secreted in the form of in their secreted form, and IgM is secreted. So we don't really know what this one does. D is like I don't know, right? So it could be involved in deleting B cells capable of producing antibodies against host tissues. So it's basically like getting rid of defective cells. But we do see its